Okay, we're back again.
Are we online?
Oh, no, we're not recording. Say whatever you want, especially if it's really personal and embarrassing.
Oh, crap, we're recording. Okay. Okay, well, we're bouncing the table now.
Oh, that's our favorite. Good. That's a feature. That's not a mistake. That's a feature of our...
Welcome to Grumpy Science. I'm Mark Tommerdahl. This is Bob Dennis. And we're here in Bob's Bat Cave, where all the great science goes on. And talking to you about today, we want to talk to you about basic experiments. Like the last episode, we just talked about a little bit about TBI, pulsar electromagnetic field experiment. And basically, how is it possible to do those kinds of clinical experiments? But let's roll it back a little bit and say, what's a basic experiment, just the simplest of experiments that you could do? Any ideas on that?
Simplest experiments? Yeah, well, those are always the best ones, actually, in the history of science, which is something Mark and I talk about all the time because we're really old and grumpy. The greatest experiments are always these really simple ones that kind of cut straight to the heart of the matter. Now, I'm not sure which one Mark has in mind, but anybody, even a non-scientist, can generally tell the profound elegance of a simple, incisive experiment. So what do you have in mind?
Oh, well, there's a lot of ways. Basically, I wanted to point out that when you do a human experiment, it gets kind of complicated because everybo's different.
I know what you're talking about. Yeah.
We're not a world full of white mice.
Mark's talking about his question was a trick question because he knows this makes me really angry.
Yeah, he gets really grumpy.
He's just trying to ruin the video, right?
Yeah, he'll start talking, and all I'll do is I'll just drink beer.
I will answer the question because I know what he means now. The simplest experiment is an experiment that you do where every single subject is genetically identical. Is that what you had in mind?
Well, and then they live in the same environment too.
So we actually have, as scientists, this is sort of one of these things, to do really well-controlled studies with a minimum variability between biological responses. We have these things called inbred strains. And we have them especially for mice, but there's also rats and there's a few other ones where basically they keep inbreeding these animals until they have a genetic defect that we want to study. And we can make transgenic animals that all, will have the same defect or the same difference. And then the idea is you're doing a study where all the animals were born basically at the same time. They've been fed the same stuff. They've, watched the same movies. Everything about them is the same. So it's like a twin study.
You may laugh, but in some of our animal experiment, I don't want to get PETA too wound up.
Well, I don't do it.
But they actually
anymore.
We don't do it anymore. They can't.
I don't like doing animal experiments, but I don't like doing them because they don't tell you the important things you need to know. And what Mark's getting at is...
Well, I was getting at the fact that we had to let them watch Sesame Street every day.
Yeah. Well, they like those things. But the problem with animal experiments is that they really, and this is a serious problem now, are extrapolating way too much from genetically identical animal experiments into completely diverse human populations. My least favorite story about this is that they were doing a drug test on a very, very promising cancer drug in a very big, prominent lab on one of the coasts. And they thought the results were so excellent that they wanted to replicate them in a different lab. So they basically took some of the mice and moved them across to a different, lab on the other coast. And another major laboratory did exactly the same experiment with exactly the same drugs, exactly the same everything, or so they thought. And the drug wasn't working very well. And they're like, whoa, what's going on? Something's really wrong. Well, it turns out that the difference between the two experiments was the mouse bedding, the little pieces of whatever the mice sleep in, was different. And when they made this bedding the same, they started getting the same results. But they argued that, well, this proves that the drug really does work as long as, everyone using it is exactly the same kind of mouse and they're sleeping in exactly the same kind of bedding and eating the same kind of food. So I asked the question, and many scientists ask the question, so how the heck does that relate to the effect of the drug in a diverse human population, all of whom presumably have different bedding, and the answer seems to be that they have taken it way too far, where they really do think that they're getting these important results, but it might be so hyper focused and specific on one type of animal under extremely narrow conditions. But you see this in FDA testing too. Many very important drugs are only tested and approved on the basis of an extremely narrow slice of the human population. And Mark and I both think that's wrong.
Yeah. No, I have.
Because it doesn't tell you anything general.
Pretty good story about that. Because this was when we first got started with our very first brain gauge precursor, the big giant beast that we showed on an earlier show. I got really excited because the pharmaceutical company was
a good story.
The scientists from the pharmaceutical company were very excited to work with us. They had developed a drug for the autism population. And this is well, I won't give the drug away. I don't want to get too many people upset at me.
And we don't want to go to jail. We don't want to pay fines. Right?
Anyway, they were very excited to work with us because what we could do is with the measures that we were taking, we were pretty sure, and the measures we'd already collected on the autism population in our lab, was that we could predict which which individuals with autism would benefit from the drug that they had developed. And basically from our studies, to me, it looked like it would be about 30% of the population.
Which is huge. It's
30% of the autism population.
Like millions of people would
benefit from this. A lot of people.
Yeah.
Well, and the scientists were pretty excited about it too, and they were all for it. And then, so I said, okay, we'll get back to us and let me know. And we were going to work with them, and I kept waiting and waiting and waiting. And I finally bumped into him at a meeting and said, well, what's going on? And he said, well, yeah, it's probably a good study, but the investors want this drug to work for 100% of the autism population. So we can't do the study because we don't want to show that it's 100% effective for 30% of the population. We want to show that it's 100% effective for the 100% of the population. That company, you know what happened to that company?
Yes, I do, but please, I don't want to steal your thunder.
They went out of business. What a shocker.
What a shocker. Well, maybe they're just bad people, or maybe they're just bad people investors. But here's the deal, okay? If you've got a drug or a device or something that's going to help a third of the people with a certain disease in a really diverse population, that's a great thing. And it's even better if you can say, hey, we can tell you ahead of time. I mean, that's basically what all this, designer, genetically based targeted drugs are all about, is like checking you and saying, okay, you're going to respond really well to this particular type of therapy. So that's a really good thing. And so the brain gauge enables us to put into different groups, different people on the autism spectrum, right? Different sorts of things. At least
we can identify different
deficits, and then different drugs have different mechanisms that are going to act on different deficits. And if they have a drug that will act on a specific deficit that we can identify, they're a candidate, right?
Yeah,
but that's not good enough for investors.
They
want the whole.
That was one of my first lessons about investors, and that's one thing that led me to. Well,
which is why we
never had an investor.
Yeah, that's right.
We'd rather just not eat.
That's right. That's right. I steal my kids' lunch money for our research, but we don't have investors. That's right. A big thing. So there we are on that.
I think that's it. I think this is our shortest video yet.
This is our shortest video.
So we can stretch it out a little longer by saying, if you're a hacker, okay. Well, the bottom line is, if you are a biohacker, you have an N of 1, and so that's kind of a perfect experiment in a way. You don't have to worry about height, weight, gender, different genetic differences, because you are genetically identical to yourself.
And here's the thing.
Or at least you should be.
In the final analysis, it matters to you whether or not something that you're doing helps you. And in a way, it doesn't matter if it doesn't help a million other people. But if you can prove to yourself that it's helping you, that's good enough. And that's kind of the power we want to put into each individual person's hands. It doesn't have to go to some big central laboratory where you pay thousands of dollars and they'll tell you from up on high, well, this has a 97% chance of helping you. You can know for yourself. You can measure it, and you don't have to take anybody's word for it.
Actually, they'll probably tell you it has an 8% chance of helping you.
Yeah, or something like that. But you know what? You don't even have to listen to that if you have the power yourself.
Yeah.
Okay, so we're done.
So, and if you suffered through our video,
once again,
don't forget there is a hidden discount code.
There's a hidden discount code that will work on Cortical Metrics, and if you're interested in that technology, we're interested in helping you out. So thanks a lot for watching.
All right, thanks for watching.